Okay, this podcast is meant to go over two concepts. The first concept is the membrane structure and function, and the second concept is going to be how things are transported across the membrane inside and outside of the cell. Some people actually already got this first part of membrane structure and function, so if that's you, then you go ahead and skip to the second half of this video. For both lectures, though, or both parts of the podcast, you should be taking notes in your spiral notebook as if you're in class. Of course, I'm going to go quicker during the video because I know that you guys can pause the video. So let's go ahead and get started. So for membrane structure and function, the things that you need to know. You need to know why membranes are selectively permeable. So when I think selectively permeable, I always like to think back to when I was a teenager and my parents would tell me that I had selective hearing. And selective hearing would be like, I would hear breakfast is ready or dinner's ready, but I would not hear please make your bed. So I would select what I would hear and what I didn't hear. I didn't hear it. So the same way, membranes select what can go through them. So permeable means that they allow things to go through them. So if it's selectively permeable, the membranes only allow certain things through them. We're going to go over the fluid mosaic model structure for the membrane. And then we're also going to talk about the role of phospholipids, proteins, and carbohydrates in membranes. Okay, so all membranes, this is always true. All membranes are barriers between the interior and the exterior of the cell, so the inside and the outside of the cell. They help maintain homeostasis, and homeostasis just means balance, so balance of pH, balance of salt concentration, sugar concentration, water concentration. They just make sure that everything is just so for the cell. They also allow for compartmentalization in eukaryotes. You guys learned last week that eukaryotic cells have membrane-bound organelles, and they have, as a result, different compartments within them. So you've got a compartment for the nucleus that's surrounded by a membrane. You've got a compartment for the mitochondria. You've got a compartment for the chloroplast, as in plant cells. And membranes allow for different sections in the cell. You can kind of compare that to your house. You have a wall that surrounds the whole house. That can be seen as a membrane, the inside and the outside of the house. And then you also have walls for your bedroom. You've got your room, you've got maybe your brother's room or sister's room, and then you've got your parents' room, and you've got the kitchen, and it's all compartmentalized by the walls. Same thing, the membranes do that for the cell. We already said that membranes are selectively permeable. So they're permeable to gas, water, entering nutrients, and exiting waste. So those things are able to transport across the membrane. But membranes are impermeable to harmful substances and microorganisms. Membranes also control ion exchange, and that's actually really important for certain areas of the body, like the heart and the brain, rely on different ion concentrations across the membrane. And they are made of phospholipids. Okay, so what's a phospholipid? A phospholipid is a big molecule, and it's made up of a head and these two little tail segments. So the membrane is actually made up of what we call a phospholipid bilayer. So you can think of this as bilayer, meaning two layers, right? Because bi means two, and it's a bilayer made up of phospholipids. So if you look at this picture down here, you have layer 1 and then layer 2. The heads are what we say are hydrophilic because they want to be near water, and the tails are hydrophobic because they want to get away from water. So the outside, let's say the outside of the cell is here, and the inside of the cell is here on the bottom. Both inside and outside of the cell are water-based. So because the heads are hydrophilic, they're going to orient themselves towards the water, so towards this outside of the cell and towards this inside of the cell. And because the tails are hydrophobic, they want to get away from the water, so they orient themselves inward. This is why you get the bilayer that looks like this. Okay, so the fluid mosaic model. So you want to understand membranes as having two like major kind of structural properties. The one is that it's fluid, meaning it's not rigid. You want to imagine a membrane is kind of like a soap bubble skin instead of a saran wrap. So soap bubble is like very fluid and like it can move well, versus saran wrap is more rigid. The membrane's more like the soap bubble, or sometimes you can think of it as like oil. And then the mosaic part of the membrane just refers to the fact that there's a lot of different things embedded within the membrane. So you've got the phospholipid bilayer, right? That's kind of like the base structure of it. And then within that bilayer, you have all these different types of proteins. You have many different types of proteins, including, I don't think they show it in this one, but you've got channel proteins. There's also these things called glycoproteins. We also have different lipids embedded in the membrane, and different carbohydrates and cholesterol. So you can think of it as like a mosaic of like many different things within the one structure. You can compare that to a mosaic painting that has many different parts that combine to form one painting. If you're not sure what a mosaic painting is, go ahead and just pause the video, type in mosaic painting into your Google browser, and you will get a picture of what mosaic looks like. Okay, fluidity. So why is the membrane fluid? Well, it turns out that it has to do with two main things. The first thing is that the tails, some of them are unsaturated fats, and the unsaturated fatty acids will kink their tails, which helps to spread out the membrane. So this is showing that here. The unsaturated, basically like the fatty acid tails are kinked, and so it allows the membrane to be more fluid. Cholesterol is also embedded in the membrane. That's this picture on the very bottom. Okay, and the cholesterol also helps to maintain the fluidity. It gives it structure, it creates more space in the tails of the membrane, which allows it to be more fluid. Okay, protein. I mentioned a few different types of protein when we talked about the fluid mosaic model, and here they are. So you've got transport proteins. Their job is to move things in and out of the cell. This is on the left side here. You see they have like a little channel that things can go through. We have enzymes are embedded in the membrane, and you guys already know that enzymes are supposed to catalyze or speed up chemical reactions. They can do this inside or outside of the cell. And then we also have signaling proteins. These are just molecules. So if a molecule from outside the cell, a signaling molecule, might attach to a protein, and it would then create a cascade of events that happens inside the cell. So for example, a cell somewhere else could send out something like a hormone, and that hormone would bind to a signal protein, and in turn that signal protein might send a signal to this new cell to do something. So this is a way that cells can like communicate with each other. Okay, extracellular matrix. We're almost done with this part of the class. So we have embedded in the membrane also glycolipids and glycoproteins. Just understand this as glyco meaning like carbohydrate. So we've got lipids attached to carbohydrates, and then we've got proteins attached to carbohydrates. These things can act as a cell label for cell-to-cell identification. So your cells need to know who's part of you and who's not part of you. And they're also important for development and immune system recognition. Part of your immune system's job is to identify things that are foreign and identify things that are part of you. So these markers that are glycolipids and glycoproteins, they allow the immune cells to know, oh, this is part of this body, so we don't have to attack it. It's an identifier. Also, glycoproteins in animal cells can act as glue, holding cells together. So there's a few different ways that they can do this. So one is through a tight junction. Okay, and a tight junction is when the cells are very tight, held very tightly together. And this would be like in your digestive tract. You wouldn't want things like acid and like nutrients just flowing out of your stomach, and or other parts of your digestive tract. So the cells in those areas are very tightly held together. We also have anchoring junctions, okay, and these are going to hold cells together like in your skin and your muscles because those body parts need to be strong, yet they need to be flexible. Your skin has to bend, your muscles have to be able to stretch, so those cells need a little bit more room to move. And then we also have gap junctions, and this is like a passage from one cell to the next for fast transport. An example of a gap junction would be heart coordination. Okay, plant cells are a little different than animal cells because they do not have an extracellular matrix. The reason why they don't have one is just because they don't have room. So the cells that are in plant cells are stacked on one on top of the other, and there's no room for those extracellular molecules. Instead of having an extracellular matrix, instead of having the extracellular matrix or the ECM, they have what's called a plasmodesma, which is a channel between the plant cells. This allows for the fast transport of water, nourishment, and chemical messages. So even though they don't have this, fancy ECM like the animal cells do, they can still communicate with each other through the plasmodesma. Okay, that concludes the cell membrane structure and function part of the class. So we're going to switch over